With the discovery of codons and the split gene, now we know that the specific gene present in two different organisms have a different structure. For example, if the gene have the same function in two different organisms, these same genes have a different structural organization in different organisms. And we also know that the same gene that is present in different organisms might have a different number of exons and introns. For example, one gene that is present in the insect genome and have the specific function, and the same gene that is present in the worm genome must have the different structural organization. Same is the case is in the humans. For example, if there is a one gene and it has the one exon that is present, first exon, but the same gene that is present in the closely related organism, that is mice, might have two exons in place of one. And it could be vice versa. In mice, there could be a one exon, and in humans, the same information, same gene could have the two exons. So these closely related genes, they are very different as regarded to the structural organization that is present in different organisms. So in 1977, the concept of split genes was put forward, and it was put forward by the two scientists, Philip Sharp and Richard Roberts. And these scientists were working on the adenoviruses. So they come up with the concept of the split genes. It means that the exons, they are intercepted by the non-coding RNAs that are called the introns. So the Richard Roberts that was working on the adenoviruses, it come up with a very interesting paper, and its title was An Amazing Sequence Arrangement at the 5' end of the adenovirus's 2 messenger RNA. And that paper was published in the very well-renowned journal, The Cell. And we know that the quality of the cell is very high. So on the other hand, the Sharp group was working on the adenovirus's protein that is known as the hexone. So the Sharp group working on the hexone, they have adopted a very unique strategy. They have taken the messenger RNA of the hexone protein, and they hybridized that messenger RNA with the hexone gene or DNA. And after hybridization, they come up with the hybrid molecules mRNA, DNA, and that hybrid molecule was analyzed through the electron microscopy. So it was very strange that the hybrid molecule mRNA, DNA, instead of having the linear formation as proposed by the linear gene theory, that molecule have the 3 loop structures. And these 3 loop structures, they are shown in this diagram. Here we can see here, this is the mRNA of the hexone that they were working. It starts from here and ends here. And they hybridize this mRNA molecule with the molecule of DNA. And as we know that in the DNA molecules, there are exons and introns, both are present. But when transcription takes place from DNA to RNA, the introns are removed in the process of transcription. So we can see here the only that part of the DNA is hybridized with the mRNA that comprises the exon. Again, the part of DNA, it hybridizes with the mRNA that is exon. Similarly, this one. And small part here. And these parts which forms a loop and do not hybridize with the mRNA, because mRNA and DNA, they are same, except the introns are removed, and instead of thymine there is uracil in the mRNA. So these three loops, basically these are the introns. Because introns are removed in the process of transcription, and mRNA, they are devoid of introns, so that's why the part of the DNA that comprises of exons, that is only hybridized with the mRNA. So this discovery leads to the phenomena of split genes, that in the DNA there are exons and then introns, exon, then intron, exon, and so on. So we have seen that after the hybridization of mRNA with the DNA of the hexon, the Sharp group, they find that the hybrid molecule of mRNA and DNA, it is comprises of three loop structures that are comprises of the introns. So after analyzing the structure of that hybrid molecule, they come up with the proposal that the hexon gene is composed of 4 exons and 3 introns, because I have told you earlier that whenever there is a number of exon, we have to -1 to calculate the number of introns. In that gene, the number of exons were 4, and we will -1, 4 - 1 will be 3, and 3 will be the introns.